So first go through with your this chapter and ask me if you have any doubt from this chapter, reproductive health. If you have any doubt in any concept, then ask me. Then we will do some questions. Basically, the questions they ask directly from the MTP and vasectomy, tubectomy, and most of the time the question have been asked from the this one contraceptive pills and IUDs, right? So these are the parts from where the regularly questions are being asked. Otherwise, it's not that much important. But yes, question have came. Go through with it. Ravi, are you going through with your NCRT?
Yes, sir.
Fine. Yeah, please go through. Done, Rabia?
Yes, sir.
Any doubt in concept?
No, sir, not really.
A question directly asked from your NCRT that which organization have developed the uh, 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 contraceptive pills Saheli? That question. It's directly from NCRT, right? So that kind of the question I've been asked. When you'll go through with the previous year question paper, you'll come to know. So let's do some previous question, right? Previous year question from this chapter, and then we'll do some practice questions, right? And I'll discuss with you some concept as well, right? So the first function, I already told you that most of the questions are directly from IUDs, copper T and all that, right? Look, this question is from NEET 2017. The function of copper ion in copper releasing IUDs. Can you tell me what is the function? So I have marked already that what are the answers. They inhibit the gametogenesis. No, they have nothing to do with the gametogenesis. They inhibit the ovulation. Look, it's a very clear answer was there. They suppress the sperm motility and fertilizing capacity of sperm. Basically, you know what happens when IUDs are fit. IUDs release copper and suppress the sperm motility, so sperm do not reaches to the fallopian tube for the fertilization. Got it? So this was the question. In case of a couple where male is having very low sperm count, which technique will be suitable for fertilization? Artificial insemination. What is the artificial insemination, Rabia? Artificial insemination, tell me, does it belong to the husband or from somewhere someone else, or from the both? Can be both.
Uh, in artificial insemination, the sperms is taken from the husband and injected into the wife's uterus
Hmm.
directly by an injection.
Right. And it can be from the donor as well. Donor can be husband as well as from someone else as well. Right?
Yes, sir.
Okay, fine. Which of the following and gamete intracytoplasmic pyramid? Uh, okay, fine. Which of the following is incorrect regarding the vasectomy? The vast difference is cut and tight. No sperm occur in seminal fluid. Irreversible sterility. No sperm occur in epididymis. Tell me. Which of the following is incorrect?
Option D.
No sperm occur in epididymis. The sperm will be there in epididymis. Look, what is the vasectomy? Try to understand first. Uh, look, this is the uvectomy. This is the vasectomy. Look, the sperm formation will take place in these seminiferous tubules, right? They will reach to the epididymis. They will try to pass through the vas difference, but at the mid of the vas difference, it is cut it down, right? So they will return it back. What will happen with those sperm which have already been formed? My next question, Rabia. They are being continuously formed. What will happen with them? They will be reabsorbed, right? They will be reabsorbed by testis itself. Got it? So,
Yes, sir.
okay. Again, come to the previous question. So, vast difference cut and type down, that is true. No sperm occur in seminal fluid, right? Irreversible sterility. Is it irreversible? No,
Yes, it's not reversible.
it's reversible. If you'll reconnect them, tube in both of the case. Look, I want to make you clear. In both of the case, right? In both of the case, tubectomy and vasectomy. If you reconnect these pipe properly, right, again it will resume their normal function, right? So this is the reversible process. Scientifically, this is
Sir, but in the NCRT, it's written irreversible.
where. Let me check with NCRT. Uh, look, it is written. These techniques are highly effective, but their reversibility is very poor. It's poor, but it's not, but irreversible. Got it?
Okay, sir.
Yeah, it means if you will reconnect them. But yes, other book like which are the good one, it is written. It is reversibility is poor. Means there are out of 100 there are 20% chance or 30% chance only, right? But yes, if you will refer the some helping book, it is written over there that they if they are connected properly, and nowadays the technology is that much uh, efficient that it's easy way. If you will reconnect them, they will resume their proper function. So it's not reversible, right? So what should be the answer? No sperm occur in seminal fluid. Sperm will not come. Why? Will the seminal fluid will come? Yes. Seminal fluid will be there. How? Look. Again, let me show you. Uh, look. The seminal fluid formation takes place. Seminal vesicle and prostate gland is over there. Right? So seminal vesicle, prostate gland, and corpus gland are there. So they will release the seminal fluid. So fluid will be there, but sperms they are coming from the where? Testis. Sperm will not reach over there. So the correct answer will be what? No sperm occur in seminal fluid. Clear or in doubt? So that is the
No,
correct.
sir.
Fine. So that will be the correct answer. It's a good question. Look, it's easy but confusing. They are the good question basically. Now come to the which of the following hormone releasing IUD? This question have been asked twice or thrice, I have seen. LNG20. So that you have to remember the name which have been given in your book that you need to remember. Right? Keep on noting on what you need to remember. So cram the load down. The cram the name of IUDs. IUDs are very important. Embryo with more than 16 blastomeres are formed due to in vitro fertilization for transfer into where? Uterus. In 2016. In context to amniocentesis, which of the following statements is incorrect? What is the amniocentesis? And basically, this is banned in India, amniocentesis. But it is not banned in many of the country, European country, American countries. Still, it's working over there. It is legal over there. What is amniocentesis and how it is done? Please, quickly, Tavia, tell me.
In amniocentesis, it is a process where the amniotic fluid is taken from the mother's womb, and it is like it is checked to see if there is any genetic disorder in the baby or
Very
not,
good, very good,
and
very good,
it's banned because it can reveal the gender of the baby,
very,
which can lead to a female
very good.
infanticide.
Right, right, excellent. Right, this is the correct explanation. Right. So, what is the in context which of the following statement is incorrect? Usually done when woman is put into 16-week pregnant, right? Because this is the time when fluid, sufficient amount of the fluid get collected, and the hormones are present over there. Can be used to detect Down syndrome. Yes, very good. Genetic syndromes are detected over there. Can be used to uh, prenatal sex determination. Yes, that is also true. Can be used for detection of the cleft plate. No. What is cleft plate? Any idea?
No, sir.
Look, in cleft palate. what happened, that the internal structure of this palate part, right? The formation do not take properly. So this is detected with the help of those ultrasound and all that, right? Not by the amniotic fluid, because this is the physiological structure. Got it? So physiological structures, amniocentesis do not reveal the physiological structure. Physiological structure means development of the organ or organogenesis, that is connected, that is the revealed by the ultrasounds and all. Fine. Uh, a hysterectomy is the surgical removal of uterus. So this question is basically out of your NCERT, right? But in 2015 it is asked. Which of the following is not sexually transmitted disease? Encephalitis, syphilis, AIDS, and gonorrhea. There are studies. Encephalitis is caused by, this is also called Japanese fever, mosquito, right? Encephalitis is caused by mosquito. It's also type of fever. Tubectomy is a method of sterilization in which a small part of fallopian tube is removed and tied up. Simple question, 2014. Which of the following cannot be detected in a developing fetus by amniocentesis? Jaundice, Down syndrome, Klinefelter syndrome. Yes. Okay. So these are the some sample of the question, and where you need to focus. You need to focus on the contraception part. Basically, IUDs. The frequently asked part is from IUDs, right? Yes. You know what is the answer of this? Answers are given here. These are the simple questions. The test tube baby technique where, okay, leave that. You can go through after the class, right? They are the simple question, but answers are given. Number of the questions are over here. Transmission to sexual intercourse. Look, you have to, what you have to remember. You have to remember gift. Question number of the time. Question have been asked from gift and not gift and zip. Yes. Look, gift, gift. Number of the time question have been asked from gift and gift, right? So you have to remember what is the gift, what is at what stage gift is done, what at what stage gift is done, right? So that you have to remember. I think everything must be clear to you, gift and all that, right? Clear to you, Rabia?
Yes, sir.
Fine. So these are the important part where you need to focus. Got it? Anything which you want to ask in this chapter? If you have any doubt, any query, you can ask. Then we'll move to the next chapter. Yes, I ask you a question. Which organization have made Saheli? Tell me. TDRI Lucknow. I think it should be there.
Central Drug.
Research Institute, Lucknow. TDRI.
Yeah.
Yeah. they have made the Saheli? That question I've been asked, already asked. Yes, Saheli. Look, it is written over here. Saheli. Pills are there. Yes, even this question. The contraceptive pills are basically made up of progesterone and estrogen, right? That are the combination of progesterone and estrogen, right? And Saheli is a non-steroidal preparation and is developed by CDRI, Central Drug Research Institute. Okay, because the steroidal preparation, like those contraceptive pills which are steroidal, they have side effect as well. Fine. So Saheli is the first non-steroidal preparation of contraceptive pills and developed by CDRI Lucknow. Okay, so these are the part where you need to focus. So you need to focus where IUDs, right, and uh, this infertility part, right. So most of the question I have seen from here. Then come to the one of the most important part of the your 12th syllabus. So this is basically what this is basically heredity and variation. Fine. So first go through with this chapter, heredity and variation, and ask me if you have any doubt. You want revision of any concept, any doubt in any concept, then we'll move to this part, previous year question paper. Do you have any idea which part have the maximum weightage in your 12th class syllabus in NEET examination, Fabia? Means which chapter have the maximum weightage?
I think genetics, right?
Right, genetics. Any idea how many question can be from genetics?
No, sir, I don't have an idea.
You know, there can be average. I like this is not from the books which I have gone through with them. Uh, this is my analysis, and I have gone through with the previous 12 year 12 yeah 12-year papers, right. So when I analyze the last 12-year paper, 17 to 18 questions are from genetics only. 17 to 18 questions. Now you can understand. 72 marks, approximately 72 or 75 marks are directly from the genetics. Then it comes to the biotechnology part, right. Then it comes to the reproduction part, reproduction in human being and reproduction in plant, right. So that is the sequence. That's how you have to take care of, right. The factual
Yes, sir.
question, concept based question, they will be from that part, ecology part, right. So go through with your chapter, this chapter, and ask me if you have any doubt. Then we will move to the this PYQ part. Is that done, Rabia? Or doing?
Yes, sir, that.
Done. Okay, fine. So first go through with some questions. So do you have any doubt? First thing. Do you have any doubt?
Sir, can you explain codominance?
Yes, sure. I want to ask you one question. Then what is the difference between codominance and incomplete dominance? Any idea?
Sir, I'm not sure, but I think in codominance, uh, both of the alleles, they both are equally dominant over each other.
Right.
And I have a doubt about the incomplete dominance. I'm not sure.
Fine, fine. I'll tell you. Look, mm, in codominance, look, try to understand. Here, the black chicken. The black chicken have its genotype C capital B, C capital B, right? And here, the white chicken, it have its the genotype C capital W, capital W. When the fertilization between these two taking place, right? So, due to the in the filial one generation, F1 generation, now there is a capital C, capital C, capital B, capital C. Means they have taken the single gene from this one and one gene from this one, right? Now a different kind of the phenotype is appeared over here. Why? Because you can see both of the genotype have been represented with the capital letter. It means both are the dominant gene. Got it? Both are the dominant gene. In codominant, both of the dominant gene will come and both of the dominant gene will try to show their characteristic. And they are unable to show their characteristic, something different is formed, right? Some different outcome is came over here. So there is a one black and blue color, right? Blue is not here. So the mixing of both of the gene, it causes the blue color. Got it? So when two dominant gene interact, both represent themselves. Is that clear, Rabia?
Yes, sir.
Now you have to remember genotypic ratio and phenotypic ratio. In F1, genotypic ratio will be 4 is to 0 and 4 is to 0, right? Phenotypic ratio. So genotypic phenotypic ratio, you have to remember the question can be asked from here. Because it have been asked, right? So it will be same as a Mendel, right? So till this point, they follow the Mendelian concept. But yes, a Mendel says this 4 will be what? This 4 will be dominant gene. Here, the dominant gene is not there. Here is something different phenotype have came out, right? So all 4 will be blue and there will be no black or white. Fine. Phenotype the same, all will be 4 is to 0. Now when it comes to the genotypic ratio, Mendelian genotypic ratio says that genotypic ratio is 1 is to 2 is to 1, right? According to the Mendel, right, the ratio will be 1 is to 2 is to 1. What is 1 genotypic ratio? Dominant. 1 genotypic ratio recessive, and 1, this is the combination of dominant and recessive. Fine. According to the Mendel, the F2 phenotypic ratio will be 1 is to 2 is to 1. It do not follow the Mendelian phenotypic ratio 1 is to 2 is to 1. It follows 3 is to 1, right? What 3? Sorry, Mendelian phenotypic ratio is 3 is to 1. Means 3 dominant, 1 recessive. Its phenotypic ratio is 1 is to 2 is to 1. 1 black, 2 blue, and 1 white. Is it clear or not? Genotypic and phenotypic ratio. That you need to remember.
Yes, sir.
Again, now come to I have shown over here another example because the question have been asked from here. Look, a person have blood group A, right? So means heterozygous A. So heterozygous A blood group IA, IO, right? Another person means male or female or mother have the phenotypic ratio heterozygous, genotypic uh, blood heterozygous B. So the B blood group is heterozygous B means capital B, sorry, IB and IO, right? So this is the dominant recessive, right? Dominant recessive combination. B is dominant, O is recessive. Here A is dominant, O is recessive, right? So when it will come to the contact, when both will uh, uh, interact, both genes will interact, what will happen? There are fair enough chances that a person can be of IB blood group, right? So A and B both will represent. So the formation of A and B blood group is the example of codominance. Here, A antigen also present in the blood group, B antigen is also present. Means both of the gene show their characteristic, right? So AB blood group is the representation of what? Codominance. So AB blood group is also example of codominance. Now I have another question. Did you got this point or not?
Yes, sir.
So what is the ABO blood group? ABO blood group is the example of what genetic incidence? Can you tell me? Any idea, Rabia?
Sir, can you repeat?
ABO blood group is example of which genetic incidence?
Uh, both of them.
Write down. Multiple allelism. What is multiple allelism? When the characteristic is one, right? So what is the characteristic? Blood group, right? So what is blood group? Blood group is a characteristic. Fine. How many genes are there? There are three genes: IA, IB, IO. When a single characteristic is represented by more than one genes, this is called multiple allelism. So ABO, why I am telling you here, because both question have been asked, right? And both question can come, right? So you will get confused, right? Don't get confused. If they ask what AB blood group, so AB blood group is a representation of, it is the example of codominance. If they ask what is the representation of ABO, ABO blood group is representation of multiple allelism when a single characteristic is represented by multiple alleles. This is called multiple alleles. Got it, Fabia?
Yes, sir.
What is pleiotrophy? This question I've been asked, but there are many concepts which are not in your NCERT in genetics. Any idea about the pleiotrophy?
It's when a single gene they um, exhibit
More than,
two or more than two characters.
right? More than two character, multiple phenotype. Very good, excellent. So pleiotrophy is also there. So codominance clear or not? So here the both of the gene will be dominant. They will interact and they will show different phenotype. Any doubt? If you have any doubt, please ask me. But you need to remember genotypic and phenotypic ratio,
No, sir, no doubt.
right? Number of the time question I've been asked: test cross and back cross. Note down, please. Test cross, back cross, very very important. Codominance, very important. Linkage, sex determination, they all are important. The toughest question from this chapter will be from where? Toughest question in this chapter will be from pedigree analysis. A pedigree is clear to you, Rabia?
So I still have some doubts in it.
Yeah. Please ask me. If you have any. In pedigree or some other concept?
In pedigree analysis.
Fine. Where is pedigree? Look, I think all symbols should be clear to you, right? Male unaffected, this blank square, male unaffected. This solid square means male affected with disease. Same way, female unaffected, female This symbol is for unaffected or unspecified sex. So what is unspecified sex? Any idea?
In which the sex is not determined. Like we don't know what
Exactly.
the gender is.
Suppose that a question says that there is a colorblind female and a normal male. They both got married and they have 4 child. Out of them, 2 child are colorblind. So child term it represent or 4 offspring. Offspring or child term is for like it do not represent. It represent their offspring, but it do not represent the sex. Four, all the 4 can be girls. All the 4 can be boys or 50% boys, 50% girl. What there can be any situation. On that time, you have to represent with this, right? Now, this line horizontal lies for mating or marriage, right? And this is slash for death and next generation, right? This is parent with the female child, parent with the one male, one female child, affected, unaffected, right? So just color bands. Yes, yes, come to over here. So the hemophilia is clear to you completely. You know how does it work? What is hemophilia and everything? You will get question two kind of the question. Either will be from hemophilia or will in pedigree. Either will be from hemophilia or will be from color blindness, and both have the same kind of. Is this concept clear to you, Rabia? Tell me.
Yes, sir.
Right. So look. For example, here I have tried to show the pedigree. For example, uh, there is a normal male, right, or normal person. He got married with a color blind or hemophilic female, right? So now what will be the ratio? So here you can see here. So like this will be XXH. So this will be carrier, right? Again XXH carrier. So both of the girls will be carrier. Means both of the daughter will be carrier. Now XHY XHY means both of the son they will be diseased, right? How we will represent? This is square, this is for the male, so this male is what? Normal male. Now I have represented female, female is shaded means this is disease. Now the next point, there are 4 offspring. So for carrier, how I will made? For carrier I have made the both of the girls are carrier. I have made the circle, right? And for the boys, I have for the sons, I have made this a square and both are the shaded. Again the question, suppose that question says that out of all daughter out of 2 daughters, one of them got married with again a normal male. What will be the ratio of color blindness in a offspring? Now how you will solve the question? Now I have made pedigree, right? So this is the daughter XXH. It is carrier. It carry the gene, but it do not have the symptom of disease. Now it got married with a normal male, right? A normal person. So normal person is XY. Again, I will make across XX. So this is the normal girl, not normal daughter. Now X XH. This is another girl is means another daughter is carrier, right? So I will say how you will represent 50% daughter are carrier. Clear? Now multiply with this one, cross with YX. So 50% son will be normal and YXH 50% son will be disease. So how you will represent like this way? One normal daughter, one carrier daughter, right? One normal son, one disease son. So handed no 50% son will be what? Here in this way in this way, this one handed 50% son will be what? Diseased. 50% daughter will be carrier. 50% of sons will be normal, 50% daughter will be normal, right? So that's how they will ask you the question. Did you got it? How the pedigree works and what kind of the question you will get. This is a story kind of the question you will get. This is the only toughest part. Tell me, Rabia, is that clear?
Yes, sir.
Always focus on the percentage. Sometime you will get confused. So, like the language, where you will get stuck. You will get stuck in the options, right? The language of options. You have to focus over there. Anything, any other concept where you want more clarity?
No, sir. All the doubts are cleared.
Okay, now come to the question. 2016 question. In human, sex is determined. It's very simple question. You know,
Yes, sir.
a gene showing codominance has, okay, a gene showing codominance has one allele dominant, another on the other, both independently expressed heterozygote, alleles tightly linked with the same chromosome, alleles that are recessive to each other. Tell me what should be that? It's easy.
Option B.
Very good, excellent. Option B. In his classic experiment on the peas, Mendel did not use sheet shape, seed color, pod length, all of above. Simple.
Option C.
Very good, excellent. How many pairs of the contrasting pairs plant studied Mendel in his experiment? Very simple. How many contrasting character he has taken?
7.
Very good. A disease caused by autosomal primary non-disjunction is. This is a good question. What should be there? A disease caused by autosomal primary non-disjunction.
Sir, I'm not sure about this one.
Which disease is autosomal disease? Tell me. Look, Klinefelter and this one syndrome, Turner syndrome and Klinefelter syndrome. Turner and Klinefelter. Both are caused by sex chromosome. What about the Turner syndrome? Turner syndrome will be X0. Another chromosome is absent. Got it? So out
Yes,
of
sir.
46, how many chromosome? 45 chromosome will be. So sex chromosome is not there. One pair of one unit of sex chromosome is not there. Clear? All autosome are there. Clear, Rabia?
Yes, sir.
What about the Klinefelter syndrome? Klinefelter, there will be 47 chromosome, right? XXY. But Down syndrome is trisomy into 21st chromosome, means autosome. So the 21st autosome will have one extra chromosome. Got it? This is caused due to trisomy. So answer will be Down syndrome. Clear? Sickle cell anemia it is also autosomal disease. And what is the non-disjunction? Can you tell me, Rabia, what is non-disjunction? Non-disjunction means at the time of gamete formation, when meiosis takes place, all chromosomes do not get separate properly. Clear? Is this clear?
Yes, sir.
Sickle cell anemia is also autosomal disease, but this is not because of non-disjunction. This is because of absence of genes, right?
Yes, sir.
Mechanism that causes the gene move from one linkage group to another linkage group?
Option B.
The mechanism causes gene to move from one linkage, right? Option B. A true breeding plant is C, one that able to breed itself. Got it?
Okay, sir.
The incorrect statement regarding to hemophilia.
Option A.
Yeah, excellent. Option A. Right. One more question. A normal vision man whose father was colorblind. This question is from pedigree. Marries to a woman whose father also colorblind. Have their first child as a daughter. What are the chances that child would be colorblind? Can you make the pedigree? This question is from pedigree. A normal vision man whose father was colorblind. Normal vision man means he do not have the gene of hemophilia. Got it? Color blindness. Clear? So XY,
Yes, sir.
normal person, right? This will be genotype and this will be the pedigree. Whose father was colorblind. Okay. Marries a woman whose father was also colorblind, right? So if a father is colorblind, what will happen? If a father, suppose that his father was colorblind, right? XY. The gene is there and there is a XX, right? So what is the chances? Every girl will be what? Carrier. It means, are you getting my point or not?
Yes, sir.
All girls will be carrier. It means the mother is what? Mother is carrier. XH. Now what will, if mother is carrier, they have their first child as a daughter. Now the first child is daughter. So let's say what's about the ratio about the daughter? XH, X. One daughter is carrier, another daughter will be normal. Sorry, XC. XCX or here XX. So one daughter is normal, one daughter is carrier. So what are the chances that child will also be colorblind? What are the chances? Got it, Rabia? That's how you have to make the pedigree.
Okay, sir.
Clear or not this question?
Yes, sir.
Pedigree question will be situational. Got it? Okay. So practice some question, previous year question, right? Go through with this all this concept from the genetics and heredity and variation chapter, because this is very important chapter. It have lots of weightage over there, right? Come with some question, come with some doubt, right? In the next class, and in next class we'll start from the next chapter, that is the this one, molecular basis of enzymes. Got it?
Yes, sir.
Okay, Rabia. Bye bye. Good night.